AS Biology. Welcome. Are you excited for a fun learning session? Make sure you have your book and a pen before we start. Today you are going to learn about proteins. Proteins. Proteins are used throughout the body in many different ways. Proteins make up some parts of the cell membranes, enzymes, hair and skin, parts of the immune system, antibodies, collagen. Proteins are made up of long chains of amino acids. The monomer is an amino acid. 2 amino acids is a dipeptide. Many amino acids are polypeptides, and proteins are made up of one or more polypeptide. Like fatty acids, amino acids have a basic structure. There are 20 different amino acids in nature, and all the different proteins are made up from combinations of these 20 amino acids. The differences between the amino acids is the structures of their R groups. Amino acids are joined together through a condensation reaction. This means a water molecule is formed. A peptide bond is formed between the C and the N. There are 4 structure levels of proteins. These are primary, secondary, tertiary, quaternary. The primary structure of the protein is a sequence of amino acid joined together by amino acids. Due to the presence of hydrogen bonds, the chain forms one of 2 secondary structure shapes, alpha helix, beta pleated sheets. The secondary structure is folded into the tertiary structure. This structure is held together by the formation of different types of bonds. These can include disulfide bonds, occurs between 2 sulfur atoms, ionic bonds. Some R groups have slight charges, and electrostatic forces of attraction can form between them, hydrophobic interactions. For proteins made of one chain, this is the final structure. The tertiary structure is important for enzymes because the structure affects the shape of the active site. Some proteins are made of more than one polypeptide chain, and these are held together by bonds. The quaternary structure is how these different chains have bonded together. For proteins made of more than one polypeptide chain, this is the final structure. Proteins have a variety of jobs within the body, and they have specialized structures to support their functions. There are a few different proteins you will need to know about. Enzymes, antibodies, transport proteins, structural proteins. Many globular proteins have a ball-like 3D structure. They are tertiary. The formation of the globular structure is similar to the phospholipid bilayer, with all the hydrophilic groups on the outside and the hydrophobic groups on the inside. Due to the hydrophilic groups, globular proteins are soluble. Examples include enzymes and antibodies. Fibrous proteins for a fiber structure. They are used within the body to build structures such as tendons, collagen in bone or cartilage. As these proteins are needed to form structures in the body, they are insoluble. Hemoglobin is made up from 4 polypeptide subunits, 2 identical alpha subunits and 2 identical beta subunits. Each polypeptide has a hem group that contains an ion. The hem group causes the red color of blood. Collagen is a fibrous protein and is made of 3 polypeptide chains wound together and held together by hydrogen bonds. Many hydrogen bonds are what help to give collagen its strength. Collagen is used in the wall of arteries to give them strength to withstand the pressure of the blood as it leaves the heart. Bones, along with minerals for added strength, tendons to connect muscles to bones. The test for proteins is called the Biuret test. The test is done as follows. Add a few drops of sodium hydroxide to the sample solution to make it alkaline. Add some copper, 2-sulfate solution. If protein is present, then the solution will turn purple. If not, it will remain blue. The color change can be subtle, so look carefully.